Our final topic in Chapter 1 is the scientific method, and you've learned about the scientific method before, and you'll hear about it again in just about every science class you'll take, even throughout college. The scientific method is a standard series of steps in which scientists use to gain new knowledge through the research that they do. So they use the scientific method as a process to do problem solving, to answer those questions that they have about the natural world. Scientists make observations, and when scientists make observations, they use their five senses. Example, use of visual sense to observe animal behavior, their sense of smell, their sense of taste, their sense of touch, so on and so forth. Instruments can extend the range of senses. In biology, we tend to use the microscope to help us see microorganisms. If you were an astronomer, you might use a telescope to see into outer space. So instrumentation can help us improve upon the observations that we make in science. And in addition to that, we take advantage of prior studies. So when a scientist does research, he looks up background information on his or her research topic to see what others have done in the field and if something that they may have used can be expanded upon or used in his or her research to help further define a particular process in which they can use to hopefully find some type of discovery. So you have learned the scientific method as a series of steps. One of those steps includes forming a hypothesis. Hypotheses are tentative explanations for what was observed, and they're developed through inductively reasoning from a specific to general. So typically how this works is you make that observation, and then you form a hypothesis, then you develop and test your experiment, from which then you draw your conclusions. And then the results are analyzed, and the hypothesis is either supported or rejected. A hypothesis is never true or correct. It's only supported or rejected. And you get that support or rejection from the data that you collect during your scientific research. Experimentation. The purpose of experimenting is to challenge the hypothesis. It's designed through deductive reasoning from general to specific. So you might start off with a very general hypothesis statement, but then you don't want to use that statement. You want to make it more general to the topic that you're testing, because you only want to test one variable in that experiment. A hypothesis often not a hypothesis. An experiment is often divides subjects into both a control group and the experimental group. And the control group is going to be the group that you use in your experiment to allow for comparison, and the experimental group is going to be the group where you have that one change, that one variable that will be changed to see the effect on the whatever you are doing. And you will make predictions then on the groups on how it should differ if the hypothesis is valid. So if the prediction happens, the hypothesis is unchallenged. If not, the hypothesis is unsupportable. The results are then analyzed and interpreted. Typically, this is the area where you collect data, put it in the data tables, and create graphs, and do statistical analysis tests and mathematical equations to show if your hypothesis was supported or rejected. And then from that data, your analyzed data to which you have interpreted, you can draw your conclusions and what the scientist thinks caused the results. Here's where you actually state, you use your data to tell the story of how it either supported or rejected the hypothesis. And we will go through this entire process and discuss data types and graphs and all that stuff, and how you label data tables and charts and figures, all in our in-class lecture. So findings must be reported in scientific journals, peer review the findings and the conclusions, and then often other scientists then attempt to duplicate or dismiss the published findings. So scientists don't always agree with one another, and they'll go out there and they'll try to dismiss published findings and say that, oh, well, I found something new here, I discovered something new here, and that could happen in science. So it's not always concrete. Science is always changing. Some things we know are fairly concrete, but there are many things that are not. Results are data, observable, objective results from an experiment. It's the strength of the data that is expressed in probabilities, the probability that a random variation could have caused the results. Low probability is less than 5% is good. Higher probabilities make it difficult to dismiss random chance as the sole cause of the result. And basically, if you recall, when we talk about the variables, you have those three variables in the experiment. You have the control variable, manipulated variable, and responding variable. Well, as we have those variables, and we'll talk about those variables in a few minutes, we also have two types of data. And you can collect qualitative data and quantitative data. Qualitative data is more descriptive in nature. Quantitative data is numerical based data. And we'll talk about those types of data collection, and then from that you could make charts and graphs. And the graphs that we use in science are a bar graph, and think about what you'd use a bar graph for versus a line graph versus a pie chart. So those are all types of graphs we use in science. And we can also build models, graphical models, representative models, mathematical models as well. So there are a bunch of different things that scientists use to show their support for a hypothesis. What is a scientific theory? A theory joins together two or more related hypotheses. It's supported by a broad range of observations, experiments, and data. Now, I want you to look up some theories of science. I want you to look up the cell theory, evolution theory, homeostatic theory, and gene theory. Look up those theories and write them down in your notebook and be able to tell me what those theories mean. What are they? Why are they important to biology? In previous science courses, if you took Earth space science, you may have learned of the Big Bang Theory. If you had a physics course, you may have heard of, or physical science, you may have heard of the kinetic molecular theory. So theories join together two or more related hypotheses. You also have scientific principles or laws, and these are widely accepted sets of theories. No serious challenges to validity. So you've heard of the law of gravity, the laws of thermodynamics, all right? So Bernoulli's principle, Pascal's principle you've heard of last year in physical science. So these are all theories that are now widely accepted in the scientific community. All right, so on to controlled experiments. In a controlled experiment, you test one variable. That would be the manipulated or independent variable. It's applied one way to an experimental group and applied a different way to the control group. What you manipulate in the experiment is going to have an impact on the responding variable or the dependent variable. And this is the variable that is going to be measured in the experiment to generate data. The two types of data that we were discussing previously in the lecture. You could be qualitative data, quantitative data. It's expected to yield different results in the control group versus the experimental group, and you should see that significant difference. But the key thing here is you only want one manipulated variable in that experiment, because if you have more than two, then you don't know if it's variable A or variable B that's having an impact and showing the differences when you compare it to your control group. So let's look. Here is a typical experiment. Observations: nitrate Fertilizers boost grain crops but may damage soil by altering its properties. When grain crops are rotated with pigeon pea, it adds natural nitrogen. So you could have two hypotheses here. Pigeon pea rotation will boost crop production as much as nitrates, and pigeon pea rotation will not damage soil. So here you can see the root nodules that play a role in nitrogen fixation in plants. So the experimental design, the control group, winter wheat planted in pots without fertilizer. The experimental groups, one winter wheat plant planted in pots with 45 kilograms per hectare of nitrate. Then the second is winter wheat planted in pots with 90 kilograms of nitrate. And the third would be winter wheat planted in pots that had grown a crop pigeon peas. All groups treated identically except for the above conditions. Here's your control group, the test pots, and here would be your data table there. So you could see year 1, year 2, and year 3, and what each bar there would be represented. What each bar would represent as far as the test pots and the control group. Experimental prediction. Wheat production following pigeon pea rotation will be equal or better than following nitrate fertilizer. The results, 45 kilograms per hectare produced slightly better than controls, 90 produced nearly twice as much as controls, and the pea rotation did not produce as much as the controls. So the conclusion, the research hypothesis was not supported by the results. However, research hypothesis was not proven false by negative results. So you revise the experiment and you grow wheat in the same pots for several generations, look for soil damage in nitrate pots, and improve production in pigeon pea pots. The results, after a second year, production following nitrates declined. Production following pigeon pea rotation was greatest of all, and after the third year, the pigeon pea rotation produced 4 times as much as controls. So now you have revised your experiment, you now have revised conclusions, the research hypothesis was supported, pigeon pea rotation should be recommended over nitrates. Sometimes studies aren't always as easily as controlled as if you were to do it in a controlled setting, such as a lab. If you're outside in the field and you're observing animal behavior, then you don't have control over what those birds are going to do. So not all science can be done in a controlled setting. And here is an example of one that you may not have any control over. So this wraps up chapter 1. We'll definitely go through the scientific method in a lot more detail through labs and homework activities. So be prepared to kind of really take in this process because we'll be using it throughout our course. And these are some key concepts that you should have picked up by the end of this chapter. So first, you should be able to define life and emergent properties and look at all those characteristics that distinguish living things from non-living things, the abiotic world from the biotic world. This is key here. I cannot stress enough that you learn the vocabulary of chapter 1 because this is foundation material that will be used throughout the entire course. You will hear some of these terms just about every chapter: homeostasis, metabolism, cell types, prokaryotic versus eukaryotic, sexual reproduction versus asexual reproduction, all those things. So learn it now because it would only make your life easier as the terminology keeps building. So good luck, everyone, and start to study.